Today I'll be doing my presentation on human immunity, and the contents I'll be talking about are as follows. So firstly, an infection is described as the body's invasion of microorganisms, including pathogens such as a virus, a small particle which does not contain an organised cell structure or nucleus. Genetic information, either DNA or RNA, is concealed inside the virus with a protective protein coating called a capsid. Only after the virus enters a living cell will it become active, and this is seen in things such as a cold, polio and HIV. Next is bacteria, single-celled living organisms existing in all environments. Bacteria is typically far less harmful compared to a virus, and DNA is contained within a bacterium in the cytoplasm. Bacteria include things such as urinary tract infection and diphtheria. Next is fungus, multicellular living eukaryotic organisms, for instance mould, mushrooms and yeast. Fungi are unable to make their own food, so they are termed as heterotrophs, and fungi make things rot in order to feed on both living and dead organisms. Fungi include things such as athlete's foot and histoplasmosis. And lastly, parasites are organisms living inside a different organism, usually termed a host cell. When a host is not present, the harmful parasite is unable to live, develop or reproduce, and parasites include toxoplasmosis and scabies. So transmission is possible by two methods: direct contact and indirect contact. Direct contact is the physical exposure to an infected person or animal. It's person to person, animal to person, or mother to unborn child, and that is often that will be done so through the umbilical cord. Indirect contact is an inanimate object obliging as a short-term reservoir for a pathogen, and these are airborne transmission, food contamination, object contamination and insect bites. Now, airborne transmission is an infectious agent lingering in the air. Food contamination is improperly handled produce, uncooked foods or unwashed salads. Object contamination is often an infected person touching something such as a door handle for a non-infected person to then touch, and thus transmitting a virus. And insect bites, they are done so through things such as mosquitoes, fleas and ticks. The mosquito is responsible for transmitting malaria. The first line of defence is the body's initial physical barriers and also the chemical barriers provided by the body to defend the body from infectious agents. Now, the non-specific response is described as a type of defence which has no specific specialisation for a particular kind of invader. So the skin provides a waterproof protective coat which is impenetrable unless it is broken. The skin is the largest of the body organs and also contains beneficial bacteria called antimicrobial peptides, which protect harmful bacteria from taking over. Next are the points of entry: eyes, nose and mouth. The tears, the nasal secretions and saliva contain an antibacterial lysosome capable of breaking a cell wall and destroying bacteria. Now, cilia hairs line each of these tracts, performing a wave-like motion of tiny hairs, sweeping dirt and debris away from the lungs. Body sweat and the skin's oil glands produce a chemical capable of discouraging the growth of bacteria. And urine also kills the bacteria as it is sterile and slightly acidic, and it is equipped with the function to clean the uterus, the bladder and the urethra, and it is then flushed from the body. If the bacteria is not immediately destroyed by the lysosome, they are enveloped inside. The mucus envelopes them and takes them to the stomach, and it is here where they are shrouded in the protein digesting hydrochloric acid, destroying the harmful microbe. The second line of defence commences once the first barriers have been infiltrated, and is done so in 3 stages. The first line and the second line of defence do not produce immunological memory. Now, phagocytosis. Monocytes, the white blood cell, undertake a variety of changes and develop into macrophages and target cells that require removal. The dead cells are submerged in a vesicle and wrapped around bacteria. This is the phagosome. Enzymes within the macrophage cell then destroy the particle that was ingested. Lysosomes, the specialised vesicle containing digestive enzymes, then fuse with the phagosome and disintegrate the substances it contains, and it is then excreted from the body. Phagocytosis is a vital contribution to the defence. Phagocytes are responsible for the engulfing and the termination of damaged, malfunction and hostile cells. As you can see, the white cell moves towards the bacterium is then engulfed, it is then digested, and the waste products that are left are then expelled from the body. Next is inflammation. Inflammation occurs following infection or at the site of injury. An inflammatory response is a range of cellular actions. A key component of inflammation is the increased circulation of blood, directed to the area affected when the blood vessels dilate. The walls of the cell develop gaps surrounding the infecting area, permitting blood cells in the immune system to move through. A plethora of cell types and proteins assemble at the site of injury, adding greatly to the inflamed area. The key indicators of inflammation are red coloration of the skin or warm to the touch, swelling due to the amount of blood at the site, and also pain due to the pressure of tissues on nerve cells. As you can see, this is an infection via a splinter. The pathogens enter the body. The macrophage is already homed here. The resident macrophage engulfs pathogens and releases cytokines. The vasoactive factors and cytokines help deliver additional phagocytes, and some cytokines initiate healing as pathogens are being destroyed. Now, cytokines are released by the immune system, and they are signalling proteins which affect the cell's communication. Cytokines produced in the embryological region openly stimulate nociceptive sensory neurons, which play a role in the trigger and perseverance of pathological pain. A key role is to activate the inflammatory response and respond accordingly when pathogens attack its host cell. Following this are macrophages, which are made from monocytes in stem cells. The enzyme released is done so by a cell organelle called a lysosome. Macrophages remove unwelcome bacteria and dead cells from the body through phagocytosis. A macrophage surrounds the pathogen, forming a phagosome around it, which then releases an enzyme inside. The enzyme release then digests the pathogen, and any residue which remains withdraws from the macrophage and is later absorbed into the body. Next is fever. A fever is caused when pyrogens, a chemical distributed by phagocytes, journey to the hypothalamus, accountable for the regulation of body temperature. Now, the normal body temperature is 37 degrees centigrade. At this temperature, pathogens thrive and they reproduce. Once the body exceeds this temperature, replication is problematic as fever acts to halt an infection. And lastly are neutrophils. Neutrophils are chemotaxis, meaning they are proficient in travelling through blood cells and are commonly first at the site of inflammation. Neutrophils surround the attacking bacteria through the means of phagocytosis and also draw larger quantities of immune cells to the site of infection by releasing cytokines. Now, neutrophils are the most abundant of white blood cells and typically fight an infection in the form of pus.